Lipids, unlike the rest of the food that we ingest, begin their digestion process in the small intestine, rather than starting the process in the mouth like a lot of the other foods that we consume. Because of this, the actual digestion process of lipids can become a lot more complex. But with the help of this video, we hope to simplify that. As macromolecules, lipids are too big to be absorbed. A lipid is made of triglycerides, but needs to be broken down into a glycerol and 2 fatty acids to be absorbed correctly into the rest of our body and our bloodstream. Once the lipid reaches the small intestine, the first process of digestion begins, known as emulsification.
Since lipids are hydrophobic, they require bile, which is secreted by the gallbladder and produced by the liver, a hydrophilic substance which helps them withstand the aqueous environments in the cell.
Bile combines with the lipids, but the chemical structure of the lipids doesn't actually change until the enzyme lipase comes in and breaks them apart. So here is our very silly and simplified demonstration. This is our triglyceride. The triglyceride is going to combine with the bile, and then the enzyme lipase is going to come in, and that enzyme is going to break apart the triglyceride. And this is the result of our broken down triglyceride. We have 2 fatty acids and our glycerol with the 1 fatty acid in the middle. Once the lipase comes in and breaks them all apart, we end up with what are now called micelles. These micelles, which contain the monoglyceride and 2 fatty acids, are made now so that they can possibly diffuse into a cell membrane with no problem. So, pretending here that the gate is our quote-unquote cell membrane, we have our micelles, which are still coated in the bile, passively diffusing into the cell membrane.
So here we have the smooth endoplasmic reticulum. The SCR's job is to sort and modify the fatty acids and the monoglyceride to then send them on their way towards the Golgi complex.
Next, the sorted fatty acids and monoglyceride travel to the Golgi body complex. There in the Golgi body complex, they are packaged along with cholesterol and fat-soluble vitamins to then be turned into fat cells, or chylomicrons. Here we have the Golgi body complex. The Golgi body's job is to then package the sorted fatty acids and monoglyceride into smaller fat cells, which will ultimately send it to the cell membrane. This all takes place in the vesicles. For the sake of this video, imagine the box as the packaging apparatus. Here the Golgi complex has completed its job in packaging the contents into small fat cells.
Next, the packaged cells leave the cell membrane through exocytosis. Once they leave the cell membrane, they enter the now easily accessible lacteals in the lymphatic duct.
So here we have our condensed fat cells, our chylomicrons, that have now gone through exocytosis and left the cell membrane. They now travel through the lacteals to the lymphatic duct, represented by this door. Once inside the lymphatic system, they are ready to be dispersed throughout the body for use. And that concludes our video on lipid digestion.